Okay. So can I start? Okay, perfect. All right, good afternoon, everyone. I'm Jasper van Eurik, and I'm here to present to you our spin-off project, XPECTings. More and more people are suffering from conditions or illnesses which makes them require donor organs. Unfortunately, the supply does not meet the demand, resulting in a significant decrease in life quality or even loss of life in general. Now, what if we could tackle this problem by, instead of aiming to replace the damaged tissue, trying to regenerate it? And this is exactly what regenerative medicine is focusing on. And one of the most promising strategies in this field is the use of biofabrication, which is basically the use of 3D printing to generate tissues. And to do so, you want to print cells according to a specific 3D design. But of course, printing cells as such is very tricky. So therefore, you combine them with bioinks, which not only make your cells printable, but also allow your cells to proliferate and be printed with high viability so that you can put them in cell culture until you have a mature culture. And once this is reached, you can use this culture for regenerative medicine applications, but also for drug screening or personalized drug screening in cancer treatments, or since you have a lot better mimic of the tissue you focus on, you can use it to replace animal testing in drug and cosmetic screening. Now, around this technology, already an entire market has emerged, and this can be segmented in different segments. So first of all, you have the companies focusing on the production of the raw materials or the polymers. Then you have companies which convert these polymers into printable inks. Then you have a lot of companies focusing on the printing hardware, companies which use this technology for custom research, and then finally, companies which use biofabrication as a key enabling technology. Now, in the field of formulations, there's currently almost a monopoly by the Swedish company Selling, and this therefore presents a gap in the pipeline, which actually perfectly matches the expertise we have at XPECTings. And we aim to fill this gap because when we look at the currently available materials, they are underperforming because they're not suitable for high-resolution 3D printing technologies, thereby being unable to form a proper mimic of the microcellular environment. They're often non-biodegradable, non-biocompatible, don't allow for sufficient biointegration, they can need toxic solvents, and mostly they are characterized by poor mechanical properties, as can be seen here, thereby limited the architectural freedom. So it is clear that there is a need for innovative materials to come up with the breakthrough life-saving solutions in these different fields. And this is exactly what we are focusing on at XPECTings. Based on decades of academic research at Ghent University, we came up with a portfolio of ready-to-use bioinks for deposition-based 3D printing, the low-resolution 3D printing, up to the very high-resolution multiphoton lithography. And this portfolio is based on 4 patents, which results in some unique features. First of all, we have the first biodegradable resin, which can be processed at unprecedented resolutions, down to 150 nanometers. We have a high-resolution hydrogel enabling cell encapsulation with high viability, a hydrogel which can be processed as a thermoplast, so thereby taking advantage of conventional 3D printing technology. And finally, we have a low-temperature processable polyester. And using these materials, we already managed to prove that they are suitable for a range of applications, including corneal regeneration. Also, we managed to 3D print a functional blood-brain barrier for drug screening, for cancer drug screening. And we managed to 3D print functional blood vessels, even going down to microcapillaries with a diameter of around 10 micrometers. And when I say we, of course, there's an entire team behind this, which is built around the expertise of Professor Sandra van Vlierbergen, which is one of the leading ladies in the fields of regenerative medicine and biofabrication. There's me as a project leader. We have a strong scientific team composed out of Agnes and Aisu, Coralie, who helps us with operational support based on her previous industrial experience, Erik as a skilled technician, and Anne to help with the business development side. Now you can see that this team is quite scientifically oriented, so therefore we aim to supplement this with business and sales-oriented people after company creation so that we can aggressively target the biofabrication market, as it is a young but rapidly growing market anticipated to reach $4.1 billion by 2026. Now this market has 2 key aspects. First of all, there's a lot of different printing technologies, which is also reflected in our portfolio. Secondly, 40% of the market is covered by the materials or the bioinks. And we aim to target this market via sales through distributors, where we already have an agreement with these 3 companies, direct sales, custom research for third parties, and organization of biofabrication trainings. Now, based on these revenue models, we drafted a financial plan where we anticipate to reach break-even by 2025, for which we need around 4 million euros of capital investment. And with this money, we want to launch the company next year, gradually expand our portfolio so that we can reach 15% of the total addressable market by 2030. So with this, I hope that I've convinced you to invest in the future of healthcare, or in other words, to invest in your future. And I'm now ready to take any questions you may have. Thank you.
So thank you, Jasper. One question, you said you're in contact already with several distributors. Mm-hmm. Are you already talking to potential clients, or which market? Maybe you mentioned it before, but what would be your target market? Who would be your perfect match on the other side? Do you already have first contacts? And if so, is it... Can you tell us a little bit about that?
Well, for the time being, we are focusing on going into sales via our distributors, which are these companies which are building the 3D printers so that we can have tailored custom inks for their printers. And basically, it's their clients who will be the end users of our products. And their clients, they can be academic researchers, but also biotech companies or companies which want to develop, for example, organ on chips for drug screening applications. But since we only started this project in January, our first strategy is to go through these distributors, as we have a complementary expertise to them.
Okay, thank you.
Happy to go next. So thanks, Jasper. This was very very comprehensive. So thanks for that introduction. I am, not very well aware of the field, but you did mention that there was one sort of supplier for bio ink that has a monopoly right now. So is this sort of a worldwide monopoly, or are there other suppliers? And you mentioned only the ones for Europe.
No, well, actually, there are some suppliers, but the CELLINK has almost a monopoly. I think they cover around 80% or 90% of the entire market, and they cover it actually worldwide. So they are the biggest player in this field. But there are some small companies emerging as well.
And does your business plan now include to displace cell ink, or are you going to add additional sort of portfolio of bio inks in order to coexist?
So our goal is indeed to coexist, because I believe we have a different focus. We to a large extent focus on the high resolution aspect, because we have some unique materials which really have some added value there. So therefore, we want to explore this high resolution biofabrication market primarily, while also, of course, focusing on the conventional market, as this is currently the largest market, and our materials also have proven very efficient or successful there.
Got it. Thank you.
Okay, so also thanks from my side. Very interesting presentation. Maybe a few questions. I think the real competitors should be companies like BASF and so on, which produce the material more than selling, maybe, or is selling really is selling not only selling the material but producing it, or how can I imagine the market?
Well, in that sense, so we have some proprietary base materials, some proprietary polymers, but going from a polymer to a bio ink still requires quite a lot of expertise there. So therefore, we are really focusing on converting this base materials, this base polymers, into a printable solution. So you can compare it with the conventional printing. If you have a dye as such is not a good ink. You need to convert it into an ink. So then you have the companies there which not necessarily produce the dye. So this is actually what we are focusing on. So not necessarily becoming the raw materials producer, but becoming the formulator of these bio-inks to make them ready to use for printing and tailor them to the specific printers.
And you have the capacity to scale this process to larger scale, because often I think in the supplier field, I realized often that there was hydrogels which were deliverable for a certain while or whatever. So you have established a process in a way that it's easily scalable to larger scale.
Yes. So we have the polymers we're using. We're already collaborating with a company where we already showed that we can produce them. Well, we already did a pilot batch at 100 kilogram scale, just to show that they are scalable. So of course, this means that this should also be scalable further. So for the production, we already have a collaboration with a specific company, and then the formulation we would do in-house. Well, we are doing in-house actually.
Okay, and the formulation you are using is printable with all kind of printers. So if you buy one from, I don't know, Selling or whatever companies exist in this area.
Yeah, so indeed, of course, depending on the printer, there can be some fine tuning. So we have currently for Regenmat, for Felix, and for Nano, we have inks which are really tailored for their device, where we can also provide the optimal processing conditions. But of course, the idea is that you could also use our inks with different printers, which may require a little bit more optimization there, but there shouldn't be any problem.
Okay.
You were talking about 4 patents, one granted and the other ones in, I think, PCT. I assume the patents belong to the University of Ghent and you're licensing them, or did you transfer the patent rights?
For the time being, they still belong to University of Ghent, and we're still currently negotiating.
What is the strategy for patenting? Are you going to focus on a special market, or do you want to go globally? Have you thought about this, or is this still far away?
No, actually, the goal indeed and the strategy is to go for a global protection, as the biofabrication market is a currently still quite a niche market. So you really have to go to a global distribution in order to get the correct numbers or the scale.
Thank you.
Maybe one question, you say it's still a niche market, but I think you have a quite let's say progressive projection on the sales over the next years. I think something up to 110 million euros per year. How did you validate this projection?
So this is based on market forecasts on the one hand, but also based on, of course, the figures of competitors. Just to see, and then just to see if this forecast makes sense.
Yeah.
And then also on the other hand, this is based on interviews with users or with people from the network to see like what is a typical, how much does an average lab typical consume per month? Is this feasible? And then taking this into account.
Yeah. So what percentage of the market you want to reach?
So we want to reach here by 2030, it's 15%, I think.
Okay.
Don't know it by heart. By 2031, we aim to go to 17.5%. But I have to make a side note there. So this is only for the part of the market which is relevant for our materials. So this is only for the printing technologies we are focusing on, and only for the materials, so not the general biofabrication market. So we already took from the total forecast of the biofabrication market, we already took only the laser-based and the deposition-based materials out of it.
Okay.
I think the 10 minutes are nearly over. Oh, okay. I do not have any more questions. Anybody, maybe?
Yeah, either.
Me neither. Thanks.
Okay.
All right, then I would still like to take the opportunity to thank all of you for your time and your feedback. And if you have any more questions, feel free to contact us through one of the channels here.
Thanks a lot. Good luck.
Thank you. Good luck.
Thanks.
Best wishes to Belgium.
Thanks.
Okay. Imagine we could predict brain biological response. It would enable a research breakthrough, opening the door to much needed treatment. This dream is now reality. Oh, shit. Everything froze. I can't pass this to the deck.
Maybe you can unshare and share again. Try this.
I'm very sorry.
No worries, please.
Okay. Can you see the full deck?
Yeah.
Okay. So I'll.
Perfect. Yep.
Yes, it works. So imagine we could predict the brain biological response. It would enable research breakthrough, opening the door to much needed treatment. This dream is now a reality. Hi, I'm Sophie Poteau, co-founder of SYNAXYS. With Michel Faring and Sandra Franconet, we constitute a team of experience and complementary expertise in R&D, entrepreneurship, and business. With a common goal, improve preclinical research on neurological biological disorder to accelerate drug discovery. Our core team is constituted of an experienced business developer and 2 scientists. We are reinforced by partnership around the development of technical assets and disease modeling. And we are supported by French institution and Disease Foundation. So today, neurological disorders are a major public health challenge. They touch 7% of the world population. They cost $800 billion every year to the society. There are no cure, those diseases are poorly understood, and the research tools are not appropriate to move forward. As a result, research in this field takes 1.5 times longer costs 30% more, and is the probability of success is less than 24%. So we target the preclinical research for the nervous system carried out by biotech, pharmaceutical groups, CRO, and academics lab. Today, pharmaceutical groups spend 2.5% of their revenue in preclinical research, with a cost growth of 10% every year. The preclinical cost for multiple sclerosis in a year is over 625 million. And there are 12 major diseases. So today, 67% of the pharmaceutical group interviewed are seeking alternate solutions. So to reverse that situation, SYNAXYS has developed 5D Brain, the first reliable human in vitro model of the nervous system. We start with iPS cells, and with proprietary protocols and media, we produce cells that are primed to assemble 3D neuronal network capable of replicating neuronal diseases. 5D Brain allow longitudinal study of those network by microscopy and electrophysiology. So microscopy allows us to identify which cells are targeted by a drug, and electrophysiology allows us to identify the effect it has on neuronal activity and the receptors targeted. The results are more informative than in vivo mouse model, quicker and cheaper. So 5D Brain is conceptually different from existing human organoids. Organoids are typically are the results of the clonal growth of standard neuronal stem cells, which is very inefficient and takes more than 90 days to grow, and the results are highly variable. On the contrary, 5D Brain production is very effective, is 4 times faster than organoids, and highly reproducible. As a result, we can reduce the use of animal experiments, we can accelerate drug discovery and drug validation by 18 months, and we can double the chance to identify sure and safe and effective drug. This enables cost saving and improve the ROE. Today, SYNAXYS operates in 2 modes, fee for service or R&D partnership. Our offer goes as 3 levels. First, pilot study, activity and mechanism determination, and identification of the therapeutic potential. We have 2 models, a healthy and epileptic one. Our revenue right now comes from service and grant. We are consolidating our IP in order to be free to engage in licensing and partnership agreement. We are constructing a database to be able to build a digital twin to our in vitro model to have a prediction capability. Our next milestones are to pose 5D Brain as a standard for preclinical test and to diversify our disease model. So to foster our development, we need to reinforce our team. We need to expand our advisory board, but also we are seeking investor and partnership to accelerate our disease model. And we are seeking partnership and collaboration to pass the regulatory habilitation to establish 5D Brain as a reference model to replace animal experimentation in preclinical tests for disease touching the nervous system. Thank you for your attention.
Thank you, Sophie. I have a question. So you're using iPS cells as the basis for the model. However, most of these neurological disorders appear in the elderly, like Parkinson and Alzheimer's. So how do you face the problem that by using your protocol, you're actually rejuvenating the model?
So that's a very good question, and we don't know yet. So we just celebrate the first year of the systems we have in culture. So we have engaged in a long-term process of monitoring the aging of those cells. We know that in 4 weeks, from an architecture point of view, we recapitulate 2 years of development, but we don't know yet how fast we can age them.
Mm-hmm. Okay. And do you have IP protection for your model? And if so, is it a method, or what is the matter of protection?
So it's a mix. So the core concept of our 3D model is a US patent that came from my past life as a postdoc when I was in Berkeley. So I'm one of the inventors in that methodology, and the other parts are part protocols and part medium. So in order to obtain that prime neuronal stem cells, we had to develop methodology to grow them, but media to drive them without forcing them to the lineage or the protocol conduce them to. And in addition to that, we had to redesign accessories and cell support to be able to monitor them both by microscopy and electrophysiology in 3D throughout the life of the system.
Thank you.
Thanks for the presentation. So maybe I go on with questions. Maybe you can elaborate a little bit. Let's say my understanding is not that you will send let's say a pre-packed plate to a customer, but that you will do it in your laboratories and send the results to the customer. Is that correct?
So right now, that's the way we operate. Because we needed to do further stability tests to see if we could be able to ship them. So for network that are only designed for microscopy, proteomics, or genomic analysis, we should be able to ship them within the next few months. But for all the networks that involve electrophysiology, we have mechanical stability issues to be able to ship them with them.
And can you elaborate a little bit on the costs? Let's say, I think a lot of work is done at academic groups, which are always, or who is always cost sensitive. Maybe you can you elaborate a little bit what you are calculating if you have, I don't know, 10 compounds to investigate them with your system?
So for a pilot study to evaluate, to map out what the problem is going to be with that compound before refining the evolution, we are less than 2K, than 10K.
Okay. And do you need any other hardware from third-party suppliers to build it, or is it in a petri dish, or how we can imagine the technology?
So the electrophysiology happened through third-party equipment.
Okay.
But everything else come from the usual base media supply and things we produce.
Okay, thanks. So NIHARIKA, you want to go on?
Sure. Thanks, Frank. Thank you, Sophie. Just a couple of questions. I think I'm trying to understand exactly what are the disease models that you have established right now, and what are the validation steps you perform. And also, if you could elucidate on where exactly you get, so what kind of, additional models do you get with the 1.5 million additional capital, or are you planning to sort of perform the validation with this 1.5 million?
So today we have a fully working system disease model for epilepsy. So the way the epileptic model was derived was based on the rat epileptic model that exists in vivo. So the level of validation is equivalent to the tests that are carried out on rats when you induce epilepsy in rat models. And we show that we get the same response in our system as you would get on rat brain slice where you've induced epilepsy. For the other model, we've established a proof of concept for multiple sclerosis. And right now we are evaluating drugs for Parkinson's disease, but we don't have a PD model. So to go back to the funding, so funding will take us to the certification and to the recognition that those in vitro model are a reference for preclinical tests for those specific diseases. And the cost, the 1.5 million correspond to a full model for one disease where we don't have preliminary data. That would be a disease where we would start from scratch today.
So new disease indication. Correct, so it would be, sorry, Sophie, I didn't get the last bit. So you would establish new model for new disease indication. I get it. And just to go back to your first point about the rat epilepsy model, so I believe you're using human iPSCs, right, to establish your 5D or 3D brain model. So how do you sort of translate from the human to the rat and then back from rat to human?
So originally the model was a rat model that was converted to a human model.
I see.
So
okay.
we have 10 years experience on the rat model before flipping it to the human model.
I see. Okay. So you still need to perform additional validation for the human model.
The human model has already been validated on the base of the rat model.
I see. Okay, great. Thanks.
So the epilepsy model is both rat and human.
Okay. Thanks.
If you look like 5 or 10 years in the future, what would your perfect business model look like? Would you have several maybe different models you can offer to test potential therapeutics for your clients? Is this kind of your vision?
Yes. So our objective is not to spread ourselves too thin. So we try to grow step by step. And the point is to have a full understanding of every disease we are getting into, so that we can have a real predictive role in signaling and understanding of those diseases, to be able to become a real partner in drug identification and development.
Great. So if some other group with a different kind of model would have an interesting model that maybe is working differently than the ones you have already, you'd be still interested in case it would fit your business model. So or yeah, okay, perfect. I think we have maybe like 40 seconds left for question time. Any further questions?
Maybe from my side, how do you want to enter the market? I think it's quite difficult, let's say, with fee-for-service to get well connected to the respective laboratories. So what's the manpower you are thinking of employ for business developers and so on?
So right now our entry has been through a research grant proposal and joining a consortium. With a mixed panel of university labs and industries, so that this model gets used and gets abused and evaluated and run through the mill, while we are still approaching biotechs and pharmaceutical groups and proposing POC and pilot studies on problems that are quite difficult. So we also interact with MDs And we get feedback from them about drugs that are actually on the market right now, for which we have access to all the data, and for which they have feedback from experience on patient to figure out what worked and what didn't work. And we are trying to analyze what we see in our system that was not seen before, to try to put that in perspective, and therefore create a new interest and show and broadcast what we can do with that system, so that we gain interest from potential clients.
Okay, thanks.
Thank you.
Thank you.
Thanks.
And how long is that? I'm on live now. All right, I will start out. I'm Anders Christensen. I am here to present APILLET. We have developed APIVault PCT, a patented enteric encapsulation platform that will be, excuse me, I have some technical issues here. Encapsulation placing other enteric platforms with a significant benefit, both the patient and the health sector as such. As you may know, enteric encapsulation is for oral drug delivery, and it protects the drug in the stomach and releases it in the intestine or in the colon. APILLET is currently looking for round A investors, and it's to support our fast journey towards the clinical phases and for strategic partners for co-development of new products. APILLET is flexible product licensing within biopharma, microbiota, nutraceuticals, oral vaccines, and small molecules, so a very wide use case. We have a commercial roadmap and a governance model in place. This is to bring the company from the R&D stage, where we are now, onto the world market in a few years. We are a very experienced team with knowledge from company management, from pharma industry, and from the production of the encapsulation. This product is further ahead compared to other projects at this business stage, we believe, because until now the focus has been solely on achieving the PCT patent. This is a unique market opportunity. The oral drug delivery market is a megatrend-driven market with an enormous market potential. It has major opportunities for sales of APIVault within biologics, for instance, in inflammatory bowel disease treatments, peptides, all GLP-1 analogs, small molecules, proton pump inhibitors, for instance, but also for the delivery of microbiota and nutraceuticals. There's a huge potential there. So even though APIVALT is an excipient in this business space, the global sales figures suggest that even a small piece of the market can become a very good business. So the novel APIVALT technology works the way that we have a two-component system. Component one is bacterial cellulose. It's a very pure, unbroken form of cellulose. It is a dietary fiber. It's not digested by human insides in the GI tract. We have a component two. It's a pH-dependent cellulase. It's an enzyme that digests the bacterial cellulose. So the cellulase activity is inhibited in the acidic conditions of the stomach and gets activated and release the drug in the neutral environment of the intestine, thus getting the encapsulation through the stomach, even though there are very varying transit times. We have in vitro proof of concept. The encapsulation is gastro-resistant. We know the compound is protected in the compound is protected in the stomach and released in the intestine. Also, we know it can pass through the small intestine and get targeted release in the colon. We have tried to model the in vivo conditions. The design space for intestinal and colon release is predicted to be, to be very wide. This is not going to be hitting bullseye with an arrow. It's going to be more like throwing a ball through an open door to get targeted release with this encapsulation. So the competition, the market benchmarks are the methyl ethyl acrylic polymers. However, they are limited with the release space and their bioactivity renders them less desirable for pharmaceutical use, and they are unwanted for use in microbiota and nutraceuticals. On the other hand, our technology has a wide design stage, so we can achieve a targeted release anywhere in the intestine, and it is made from material generally recognized as safe. So there are no marketed competitors with these features as far as we know. COGS will be volume dependent, of course, the cost of goods sold, but we expect to produce high volumes and therefore reach low COGS over time. At present, APILLET has 6 projects in the pipeline, 4 of which are built on the APIVALT technology. So this is a unique value proposition, a wide range of use cases, targeted application, and a solid innovation pipeline. We will follow our commercialization roadmap and will continue to develop new innovations within the field of oral drug delivery. And the business model that is built around licensing of our encapsulation and product development services. So our ask is around A. We are going towards producing a prototype. We're going towards preclinical trials and commercialization. 3 million for prototype and preclinical trials. 1 million for business development and commercialization. We need to get out to a lot of different use cases. To keep this together, we need to build an organization. So this was the very short version of the visions for APILLET. So please, if you have questions, I will be happy to answer.
I'm happy to go first this time. Yeah, thank you very much for that pitch. Just one question on component 1. So you said that it's a bacterial cellulose and that the product is generally safe, but what is the degree or the risk of immunogenicity with a bacterial cellulose component?
Bacterial cellulose is as any other cellulose, and we eat a lot of cellulose every day. And immunogenicity with cellulose, I will say that is non-existent. It, of course, has to be investigated. I know it has been, bacterial cellulose has been used for many years in Asia as as a supplement for pectin, I think it is. Well, anyways, it has been used as a supplement. It's been used many years as a food. So I don't think there's any problem there. There could, of course, be a problem with the cellulase and immunogenicity, and that has to be investigated. I don't think it's going to be a problem. There's a lot of cellulases out there, and I would be very, very surprised that if there was any immunogenicity around there. And also, you should also add that there's a lot of cellulases commercially available out there. They have been studied widely to be used for biofuels for many years, and so they are very well known. Also, the enzymatic reactions around the cellulases.
Great, thank you. Just another short question. So your pipeline right now, the clinical pipeline, are you planning to go for a 505(b)(2) regulatory path, or are these new agents that you're encapsulating?
I mean, the regulatory pathway will be decided by the partner, the strategic partner, because we cannot say about that. We are open for any suggestions if it is viable commercially, definitely, and that is both within the human health space and also animal health space. It might be attractive with animal health because it might be a faster way to the market.
Okay, thanks.
Yeah.
Thank you.
So if I go to the pharmacy, there are plenty of microbiotics. What's the problem right now? Why did you feel you need a better solution? What is your USP? That's not my field, so maybe you can give us some insights.
No. The competition, that is the methyl ethyl acrylates, which have been used for many years. They cannot be used with the microbiota and nutraceuticals because they have EMA and FDA. They only approve minor intake of these copolymers, and because they do have bioactivity, I suppose. Also, they have been for years. The ethyl acrylate was observed by the FDA because of carcinogenity in rats. They cause irritation, skin and eyes. So also, just in my view, these. I mean, if you want to give a pharmaceutical, you don't want something which do have bioactivity inert within it. So I think that is basically the edge that we have. That we have a very wide design space. We can target the release many places in the intestine and in the colon, and it is made from grass materials, which will make it easier for us to get it through the regulatory approval.
Thank you.
And let me also add, of course, that it is we have a patent on this, and there's no patent on any of the other encapsulations. So that will also mean that you will get some kind of patent protection from the drug that you are actually giving in this encapsulation. I also see that as an advantage.
Is the IP in your startup, or is it with a academic institute?
No, it is in our startup. Yes.
Okay, thank you.
So one question would be: you said that you need the seed funding to generate the first prototype. So this does mean the current status of the development is that you have the polymer developed, but you don't have so far any real proof, for example, using insulin or whatever to test the technology.
What we have used until now, that is in vitro experiments, and we have used a model drug, which was not a drug as such. It was a compound that was easy to measure and easy to follow, and how it behaved in the encapsulation. So that is what we have at the moment, So we haven't looked with the real drugs yet.
Okay. And you mentioned that the goal would be, for example, to encapsulate also biomolecules like antibodies. Is there any proof of concept that this could really work, not only with your technology, but in general, that this would be a good mode of administration?
No, not that we know of. We have not. I mean, we just mention it as one of many use cases. It is. I mean, inflammatory bowel disease. I know there are projects that are trying to target intestines with antibodies or with some kind of fragments. But I do not know the current state of that. And I think also maybe the issue here is that if they have issues getting it into the intestine, actually. So so that is, please go ahead.
So what and who would you see as your main competitors in this field?
There's a lot of producers of the methyl ethyl acrylate copolymers. Because they dissolve when they reach pH 5.5, pH 6, but they are, as I said, it's the same. Plexiglass is that is what people are eating when they eat this, and so it's not the best solution, but it is the solution, and so at the moment, as I see it.
Okay, thanks.
Did you already talk to any microbiota or nutraceutical companies if they would be interested in your encapsulation?
Yes, we have had a little bit of discussions with different companies, and also, we have had discussions with a number of different players in the field, and there is definitely a lot of interest in this. It is very much, I see two things with this technology. It is very much out of the box. So this is not something anybody has used before. That is also why we have been able to take the patent. It is because it's not covered this field. So that is also makes it also early days for this project. But in the other end of this project, there will be a great award if we can really make this work. And I believe we can, because this is, from now on, it is going to be engineering work. There might be bumps on the way, I'm sure there will, but they can be overcome.
So we've got 1 minute left. Very short question on my side. Is it, I mean, for anything you have to swallow, the taste is quite important. How does it taste?
I haven't tried it.
Is it
no, but you know,
neutral?
Yes, it's neutral. It is. I mean, it's somewhere between paper and wood. That is the kind of that's also the texture of it. So. It is true. That is actually not true. I have tried it. It was an Asian version where they had used it as what is that called? Fruit gum. Maybe it's called fruit gum in English. A sweet. And then there was artificial flavoring. There was sugar in it. It tasted great.
Okay.
So you can of course also put this in these encapsulations. You can get it with any type of added flavor.
Okay. Thank you.
Thank you.
Thank you.
Thanks a lot. Bye.
Bye bye.
Bye bye.
Bye.